Good morning, students. Today we will learn about vegetative reproduction. This vegetative reproduction can also be referred to as vegetative propagation. This is a method of reproduction occurs in plant in which new plants are produced from various parts of parent plant, such as roots, stem, leaves, and bud. Vegetative reproduction is a type of asexual reproduction, occurs with the help of vegetative parts like stem, roots, leaves, and bud. It may take place naturally or artificially. So, there are two types of vegetative reproduction. First is natural vegetative reproduction and second is artificial vegetative reproduction. First upon, we discuss about natural vegetative propagation. There are many plants which propagate naturally. Some plants propagate by roots. Examples are sweet potato, guava. Some plants they are propagated by stem like ginger, banana, potato, etc. Some plants are propagated by leaves like bryophyllum, begonia, etc. Natural vegetative reproduction takes place without human involvement. Natural vegetative reproduction by root. In some plants, root becomes swallowed due to the storage of food materials. Such roots are called as tuberous roots. There are three types of tuberous roots. First is a simple tuberous root. First type of tuberous roots is a simple tuberous roots. In simple tuberous roots, the roots is single. Example are sweet potato, dahlia, etc. Second type of tuberous roots is fasciculated tuberous roots. When these roots are present in a group or cluster, then it is called as a fasciculated tuberous roots. Example is a asparagus. It is a medicinal plant. In Hindi, it is called as a shatavari. Third type of tuberous roots, it is nothing but a non-fleshy roots which help in vegetative reproduction. Examples are guava, muraiya, etc. Muraiya means it is nothing but the kadi patta. Now we see natural vegetative reproduction by stem. Example is potato. In potato plant, tuber is a much thick underground part of stem to store food and carry reproduction process. Example of natural vegetative reproduction by stems are ginger, turmeric, potato, etc. Now we see natural vegetative reproduction by leaf. Examples are bryophyllum, calancho, begonia, etc. Now here, in natural vegetative reproduction by leaf, new bryophyllum is developed with the help of leaf. These leaves are nothing but the bird-like structure. With the help of leaves, number of plants' body are developed, and this is called as a natural vegetative reproduction by leaf. Now we discuss artificial method of vegetative reproduction. Artificial method of vegetative reproduction, also called as horticulture. Horticulture means practice or growing of flowers, fruits, vegetables, etc. Artificial method of vegetative reproduction is done by the involvement of human being. It is carried out in the plants which do not produce seeds or produce less number of seeds. There is no change in the genetic makeup. Basic types of artificial vegetative reproductions are cutting, grafting, and tissue culture. So, this is a picture of cutting. This is grafting method. And third type of artificial vegetative reproduction is tissue culture. So, first upon we discuss about cutting. In cutting, the small pieces of the plant parts like roots, stem, or leaf are placed in moist soil and new plant is developed. So, stem cutting is used in rose, leaf cutting is used in sansevieria, and root cutting is used in blackberry. Now we discuss second type of artificial vegetative reproduction is grafting. In this method, parts of two plants are joined in such a way that they grow as a one plant. In grafting method, rooted plant called as a stalk, while over other which is inserted on the stalk called as a scion. Now here we see third type of artificial vegetative reproduction. It is nothing but tissue culture technique. Tissue culture, artificial vegetative reproduction is also called as a micropropagation process. In this method, first we selected to the mother plant. From this mother plant, we separate apical meristematic tissue from the apical region of plant. We selected apical region because apical region is free from infection. This apical meristematic tissue are the parenchymatous tissue. Now, this parenchymatous tissue we wash with the alcohol to remove their infection. Now, this tissue we put into the growing medium. This growing medium called as a agar gel, which is prepared from the red algae. In growing medium, there are plant growth hormones are present. These plant growth hormones stimulate the growth of callus and number of seedlings are produced, so that due to the tissue culture technique, large quantity of plants are produced by the tissue method. Tissue culture is also called as a micropropagation, by which large number of plants propagules are produced. Now here we discuss about advantages of vegetative propagation. The first advantage of vegetative propagation is it is a rapid process, it is a cheaper method and also easy for reproduction process. By the help of vegetative propagation, the plants they are genetically identical. Here, superior quality of fruits or flowers are going to maintain by grafting process. New plants produced by vegetative propagations are disease-free and also there is early formation of flowers and also the fruits. Along with advantages, we also discuss the disadvantages of vegetative propagation. The first disadvantage of vegetative propagation is new varieties of plants cannot be produced by vegetative propagation. Many times, plants developed through vegetative propagation have short lifespan. Third, it is more expensive as compared to multiplication of plants developed from seeds. It requires trained, skilled, and experienced person to carry out processes like cutting, grafting, budding, etc. Now here we discuss one question. See, do you know why does gardener choose to propagate plants asexually? Gardener choose to propagate plants asexually because asexual propagation is an easier and faster method. Also, they preserve genetic characteristic of the plants and also they maintain their genetic materials. Thank you.